Hello everyone, my name is Fayaz Khan Sahil and today we're gonna learn about diffusion, osmosis and active transport. Let's begin. What is diffusion? Diffusion is a net movement of solute from an area of higher concentration to an area of lower concentration down the concentration gradient. So what does this line mean, from higher concentration to lower concentration down the concentration gradient? Let me explain you by giving you an example. Suppose these are like perfume molecule. Okay, so you can see the perfume molecule. They are in higher concentration, right? So as they are in higher concentration, this molecule doesn't stay in same place. They actually move, right? They actually move. No molecule stay in same place, such as gas, right? Solid stays in same place but not gas, right? So these gaseous molecules moves from higher concentration to lower concentration down the concentration gradient. So diffusion doesn't require energy, so it is generally moved by this movement of the particles from higher concentration to lower concentration. Now let's look at factors that affect the rate of diffusion. The first factor is temperature. As temperature increases, for example, the particle movement is this, right? Let's take a hypothetical value, okay? Suppose this is 15 meter per second. They are traveling at 25 degrees Celsius. This particle X is traveling at this speed. But as you increase the temperature, this particle now move at a higher speed. For example, at 60 degrees Celsius, this particle moves 60 meter per second. So the more you increase the temperature, the higher the movement of the particle. And the rate of diffusion also depends on the size of the particle. When the size of the particle is small, the movement is generally faster and the rate of diffusion is higher. So if a particle is larger, for example, this is a particle, this is a particle, so it has higher mass, right? As a result, this will diffuse slower than this particle X, and this is Y. So as you can see, the size of the particle is also important for the rate of diffusion. Now we have surface area. For example, when the surface area is small, the particle is spread out faster compared to when the surface area is large. For example, suppose in this container we have let's take four molecules, and let's take another container. Let's just take another container with same four molecules. Now tell me which one will diffuse faster. Suppose this is 1, this is 2. If your answer is number 1, you are absolutely right. This is because when the surface area is small, the particles spread out faster. For example, how much time it will take for the particle to spread out this much, right? Over here, you can see the distance is much larger than this one. That's why the more the surface area, the lower the rate of diffusion. If the surface area is small, the rate of diffusion is higher. Okay. Now let's look at another factor that affect the rate of diffusion, that is concentration gradient. For example, if the concentration gradient is steeper, then faster rate of diffusion happen. Okay. For example, let me give you guys an example so that you guys can understand it very carefully. What does this mean? For example, over here we have 12 molecules. Okay, over here we have one molecule. Over here we also have 12 molecules, but we have over here almost 6 molecules in that side. So which has steeper diffusion gradient, steeper gradient? Of course this one, because you can see there are 12 present, 12 molecule is present. Over here is only one. Over here it is only 12, and 6 is present over here. So as you can see, A has higher steeper gradient. And also distance. Okay, this refer to the thickness and thinness of the cell membrane. Shorter the distance, faster the rate of diffusion. For example, as you can see, this is a cell membrane. This is the distance is very shorter, right? So higher rate of diffusion. But if it get larger, the rate of diffusion will be slower. Now let's look at another topic, which is osmosis. Osmosis is the net movement of water molecule, or we call solvent. Why this is solvent? Because most of the chemical reactions occur in water, so it's a good solvent, right? Most of the substance also dissolve in water. So what is osmosis? Osmosis is a movement of water molecule from a region of higher water potential to a region of lower water potential. This movement is by partially permeable membrane down the water potential gradient. You might think this sounds so complicated, but it isn't. Let me explain you. For example, this is a cell membrane. Let's take this is a cell membrane. Okay, let me draw some water molecules. Now water molecule should be blue. Okay, this is water molecule. Okay, and over here we also have water molecules. So as you can see, over here we have almost 10 water molecules, right? 10 water molecules. So over here we have only 4. So as you can see, it has higher number of water molecules, which we call higher water potential. Higher water potential simply mean higher number of water molecules. So it moves from higher water potential to lower water potential down the concentration gradient across a partially permeable membrane. This is partially permeable membrane. So what is partially permeable membrane mean? You should ask me that question. Partially permeable membrane. So partially permeable membrane simply is it doesn't allow all the substance to pass through it. It allow only specific substance to pass through it. So it is very specific. Partially permeable membrane is very specific. It allows only specific substance to pass through it. That is what partially permeable membrane is known as. So let's look at the effect of osmosis on animal cells. This is very important for the exam, by the way. We'll learn about 3 type of solution: hypotonic solution, hypertonic solution, and isotonic solution. So let's look at the effect on the animal cell. Let's look at hypotonic solution. The hypotonic solution has a lower concentration of solute, more water compared to the inside of the cell. You might think this sounds complicated, but it isn't. Hypotonic solution are the solution which has more number of water molecules than inside of the cell. For example, this is a hypotonic solution. For example, and inside there is only this amount of water molecule present. So hypotonic solution has more number of water molecules than inside the cell. So what did we learn at osmosis? Water will move from higher water potential to lower water potential, right? Down the concentration gradient. Okay, so when all of this water molecule entered the cell started to increase in size, and eventually it blast. As you can see over here, it actually blast. And when I was in O level, okay, I was always confused between hypotonic solution and hypertonic solution. I always used to mess this up, so you should not mess this up. So it took me a while to get that in my brain. So that's the thing. Oh, by the way, I don't know where you guys are from. Please write that down in the comment section, and please do subscribe. I'm really new to the YouTube thing, but I'm really excited to teach you guys. So water molecule, let's look at the observation what your sir has made. Water movement, water enter the cell by osmosis. Effect on animal cell. The cell swells, as you can see, cell increase in size, then it blasts if too much water entered the cell. Now let's look at hypertonic solution. Hypertonic solution has a higher concentration of solutes, which means it has less number of water molecules in the solution, okay, than the inside the cell. So now it is Uno reverse. I hope you guys play Uno, right? So Uno reverse, over here the cell has more number of water molecule than outside of the cell. As a result, what is the movement of water should be? As we have learned the definition of osmosis, it is water move from, guess that is right, higher concentration to lower concentration down the concentration gradient. Sorry for my handwriting, I know it isn't that much good. I'm not used to write in a graphics pad. So as you can see, so what happened to the cell? The cell, look, this is red blood cell, we commonly known as RBC, okay? RBC isn't like this shrink stage. This is the final result what happened. RBC is just like this biconcave shape, okay? Or you can see in this position. So it used to be like this shape, okay? But what happened is that when water started to go outside of the cell becomes shrink to this condition. So let's look at the text. Water leaves the cell by osmosis. The effect, cells shrink and become crenated or shriveled. So you can see the cell actually shrinked, right? Isn't that interesting? Now we look at the other interesting topic, which is isotonic solution. What is isotonic solution? Isotonic solution, basically, if you ask me the definition, the concentration of solute is equal inside and outside of the cell. You can simply say the number of water molecule inside and outside the cell is equal. So there is no net movement of water molecules. So what will be the movement of water molecules? So for example, if let me give you guys an example. So if 10 water molecules move outside of the cell, 10 water molecule will go inside of the cell, okay? So what is the effect? The cell remain normal in size and shape. The cells should remain normal in size and shape, right? So as there is no net movement of water molecules. Now let's look at the final diagram. As you can see in hypotonic solution, let me zoom it out for a bit. As you can see in hypotonic solution, the cell actually blast because number of water molecule was higher inside the cell. That's why it move inside the cell increase in size and blast in hypotonic solution. In isotonic solution, as you can see, there is no net movement of water molecules, so the cell remain the same size. Let's look at hypertonic solution. Hypertonic solution, there is more number of water molecule present inside the cell, so water move from higher concentration to lower concentration down the concentration gradient to outside of the cell. Now let's look at the same thing will happen when a plant cell is kept on hypertonic solution, isotonic solution and hypotonic solution. Let's look at what happened in hypertonic solution. The cells shrink, as you can see the plant cells shrink. This is by the way cell wall, okay? Cell wall is the structural component of the plant, as you guys should know. So what happened in isotonic solution, there the cell remain same in size, and in a hypotonic solution, cell the water molecule get inside the cell. But you should ask me why this cell didn't blast and animal cell did. This is because of the cell wall, okay? The cell wall is a structural component that provides structural support. For example, this is a cell wall. The water molecule are providing, so for example, the water molecule are providing pressure on this area, right? So what will happen is that the cell wall will give equal amount of pressure. As a result, the cell doesn't blast. Did you guys got it? Now we have one more topic left, which is active transport. So what is active transport? Active transport is a movement of dissolved molecule in and out of the cell through the cell membrane from a region of lower concentration to a region of higher concentration. The particle move against the concentration gradient using energy released during respiration. Let me give you an example through this molecule. For example, plant cell need nitrite ion, right? For example, this is outside of the cell. For example, this is like nitrate ion, okay? This is nitrate ion. But if you count the number of nitrate ion, 1, 2, 3, 4, 5, 6, 7, 8, there is 8 molecules of nitrate ion and there is only 4. So in theory, this nitrate ion should move in this direction, right? But plant actually need nitrite ion, right? So it actually absorbs nitrate ion so that it can utilize later on by using cell energy. For example, it actually uses ATP. ATP is like batteries of cell which provide energy. So it uses ATP to move this nitrite ion inside the cell. As you can see, outside of the cell it is lower concentrated, right? Lower concentrated. So it moved from lower concentrated to higher concentrated against the concentration gradient. Why against? Because the gradient should be higher to lower, not lower to higher, as in active transport. That is why it is called against the concentration gradient. So as we have learned from this, so these molecules are actually moved through a carrier protein. I hope you guys learn all of the lesson very carefully. See you guys in the next class. Please do subscribe to the channel and let me know which topic you guys wanna know. I'm gonna cover each and every topic. Goodbye for now. Tata.